everyone for joining us for this Tools for Tomorrow special edition. We're gonna have all three BRCs give you an update on what we've been doing as it relates to Hantavirus, and Philippe from Broad PDN is gonna go first.
A good way to start to understand the biology of a virus. We have in PDN the VirZone resource that describes knowledge about all kinds of viruses. And here you can find, if you look by a taxonomic search in VirZone or just by the search, you can find the ortho-hantavirus, which describes how is the biology of this virus. You see the genome as 3 segments. It's a negative strand RNA virus, segmented, and encodes for 3 proteins. One of them is cleaved in 2 parts. This is the glycoprotein. So it's pretty simple virus compared to corona or pox virus, actually. And you have some information like there are actually 2 enzymatic activities, so and the way it replicates and so on. We made for the outbreak resource a special, more detailed data into VirZone with a fact sheet dedicated to Andes virus, because the precedent was for the whole genus. We'll see that there's an extra protein in the genome with a link to the reference strain, which is a Chile 1997, and the new strain, which is in Pathoplexus here. We have some news about, some data, about the proteome, the variant that displayed of the outbreak, and some hantavirus diversity. So I told you in the ortho-hantavirus andesis, so this is the name of the species of Andes virus, we displayed the genome with an extra open reading frame called the NSS that some hantavirus encoding for. And we also made a more detailed picture of the variant, especially the glycoprotein, which is cleaved in 2. Here you have the topology in the proteome part. You have access also to cell receptor, which is published. It's access to UniProt or to Wikipedia. And all the protein can be accessed in these databases. We have also some analysis on the first sequence of the variant from the outbreak. We mapped that on a 3D picture of the glycoprotein in tetramer. It's a tetramer. So you see it's pretty much all around. And there is not much mutation for a virus compared to the reference, which is 1997, 30 years ago. You see few mutations actually occurred, which is kind of a relief. It means there's a lot of pressure for kind of human adaptation or anything like that. Here you have also the topology of the, like this is a signal peptide, the transmembrane and so on. You see where are the mutations in the sequence of Switzerland. Also, some details on the diversity of hantaviruses. There are many hantaviruses, many old world hantaviruses and new world, which doesn't induce the same pathologies. And this virus is here. It's all hantavirus are adapted to rodent host mostly, and a specific one host. So you have many of those. You have 52 species of hantavirus actually, and they are all dedicated to special hosts. So Andes is really for a special rodent host in the South America. And I told you there was linked to Pathoplexus. So Pathoplexus in PDN is a platform to share the sequence for everybody, and they have very quickly made Andes virus part of it. So you can see if you click on it, you have a lot of sequence. We have 500 sequence. Among them, 8 are from the recent outbreak. You see the sequence in Netherlands, Switzerland, South Africa, Canada, France. In this, each entry, you can have the metadata in the first part and link to ENSDC. In the lower part, you get all the substitution compared to the reference strain I told you before, Chile in 1997, in terms of nucleotide or amino acid with the FASTA sequence. So you can reuse the data, and there are some restrictions for data that are not being published, so you can use it, make on site, on presentation, but you cannot publish it unless you get authorization. And also the third resource of PDN is in Nextstrain, which is linked to analyzing the phylogeny of viruses. They also made very quickly an Andes Hantavirus subpart, which is per segment, because of course you have to compare the same kind of nucleotides. And so this is the M segment coding for the glycoprotein. And you see you have a map with some sequence in Europe, South Africa. These are for the outbreak. And the endemic sequences here. We see that we have some red sequence, so the outbreak is here, and you have some sequence from Switzerland in 2016, and it was actually two travelers that went to Chile and went back to Switzerland with the virus, which as well, just two cases, and it was described already. So you can see the topology of the phylogeny of this segment. You can change the segment by change data set here. For example, you can go to the L segment. This is it. We have also the same kind of analysis for the three segments. Okay, that's all for PDN resource, and thank you.
See, here there's a timeline and a picture of the boat, but that covers most of the the details and the history here of the different cases. It started probably in the beginning of April with sort of a rather large gap of no recorded transmissions. Currently there is, so I think we have 10, potentially now 11 probable or confirmed cases. We haven't seen a lot of news in the last few days on this, but I'll have a summary of that a little later. Yeah, so the Swiss lab had the first sequence, but what I maybe wanted to point out that there was a very nice joint virological post by the 4 countries that have so far produced genomes, which is up on Biological, as I just said, sort of coordinated by Gustavo Palacios. And that is a nice example of a collaborative, multi-country initiative to analyze these data. As Philippe says, these have been shared on Patoplexis. It was quite nice to see that Patoplexis was able to spin up this instance in about 15 hours of work from the time that they had decided that they want to do this. And all publicly available sequences from the outbreak, we believe are in the database right now. Yeah, so we have both these Nextstrain link outs. So that's Philippe showed this build on Nextstrain that was made by Emma, but there's also sort of tools integrated into Patoplexis where you can any new sequences that get submitted, you can immediately send to the Nextstrain tool here, analyze it in the context of existing genomes. And last piece, yeah, I wanted to give a shout out to Ian McKay, who provides these sort of very nice graphics of the timeline of the outbreak and so forth. So there's, I can provide a link to this, but I think it's a very useful summary of all the different news in all the different countries that have experienced this outbreak. Yep, so that's it for me from the Hantavirus.
Broadly, just wanted to quickly let you guys know that we have some very recent updates to the resource as a whole. So we have this priority pathogens tab added, which has a list of priority pathogens to review and download and use for your whatever you would like. We have family level phylogenetic trees on every family page, so those are also able to be viewed and downloaded. And I'm going to spend the most of the time looking at Archaeopteryx today to see to get the most out of our phylogenetic visualizations. And then our viral assembly service has been updated to make the IRMA modules more clear. And then also we have added reference guided assembly, so feel free to use that as you're assembling hantavirus genomes or any other virus that you are interested in. Jumping into hantavirus specifically, we have this outbreak phylogenetic analysis report, and I'll show you how to get there from the website, but that has information for you to read through, and then the trees that we have generated, and we also use that awesome virological source that Richard just mentioned. So here's an example of one of the trees that we generated. and you can see that this is the sequence that we have in this tree is the one that is in sync, but definitely use all of the sequences that are in Pathoplexus and get the comprehensive view of the outbreak from Argentina. And they're unlikely to have reassortment since they are pretty much the same. And so those trees for each segment live in this workspace, which I'll also show you. And in that workspace, there is the tree file, and then these additional files that have the align FASTA, the metadata used to generate the trees, a number of different tree files for downloading and viewing, or looking at further in the BVBRC. And I also just wanted to mention some BVBRC tools that you can use to analyze data. So as it pertains to outbreaks or just analyzing data in general, we have a number of tools and services that you can run on any publicly available data or your own personal data. Some ones that I thought would be important to highlight as it relates to an outbreak is our whole genome SNP analysis service that uses KSNP4 to identify SNPs and estimate phylogenetic trees based on those. So you can use a BVBRC genome group to generate those. We already talked about the viral assembly service. The viral genome tree can take in that genome group, FASTA aligned or unaligned, and then you can set whatever parameters as you run that tree. And then we also have multiple sequence alignment and SNP variation analysis service that can start with unaligned or aligned sequences from a number of different inputs. We have a few different aligning methods you can use for that. But I wanted to jump to the BVBRC so you can find this data. One thing that's important to note is the links. So you can see the BVBRC and you can see the data, but anything running an analysis job or files in a workspace, which is where this data is kept, you must have a BVBRC account to use it. And that is very easy to do and free. Just know that if you're like, wait, that link didn't work, that's just because you don't have a BVBRC account. So this lives in our news and announcements for Hantavirus 2026. And so you can click that and that will take you to that page that I showed you, which has that summary and the report. And then also the method for how the trees were generated. Christian Zamasic built this tool, VFAM trees, that is generating these trees very quickly. So we're very thankful for that. But in the results section, you can see the link to that workspace that I mentioned. And in that workspace, we have the segment trees. And if you click into one of those, have the tree file and those additional files. When you open the tree, this is where I wanted to just spend a few minutes talking about the functionality of Archaeopteryx, which you can also read further in this user guide. But you have the ability to display any number of the data. So I'm going to remove the internal nodes and the sequence, but you can remove and put them back and add things as you're interested. And then the zoom function also allows you to show different things. So you can zoom in vertically, which would expand horizontally the same this way, but then this F will fit and center the tree to your display. So it's a really good place to start to get the whole tree on one screen. You can also search for a specific metadata feature of interest. So if I wanted to highlight Homo for Homo sapiens. That would highlight all of them, which isn't the most helpful for this. But then you can also do 2026, which will put that Switzerland sequence up. And then you can also change the metadata labeling color, so we can color by year. That will add those in. And then you can download this tree image in a number of different formats. If you just want the image to use for something further, then you can download that. And all of these things can be changed to make it more visually interesting for you. And that is, I think, the only thing I wanted to show you with that. I will hand it over to Anton.
Okay, so yes, thank you. Actually, before I start, since I have exactly 70 seconds, I actually have a question to Patoplexus guys. Do you guys have NCP server for all of these things, or are you planning to create one? And the other question is that are you planning to move these things to NCBI, I mean, or EBI to have official identifiers?
So I guess these were two quite different questions. I don't think we have a plan to add an MCP service at the moment. This has not really been discussed. Regarding moving these data to NCBI, so this would either happen after a year or whenever the authors of these data switch them to open data use terms.
Okay, thanks. Okay, please, next slide, please. Okay, so we are a bit of a different resource. We don't provide pre-computed things. Well, we do some, but mostly we provide this pathway from data to inference. So BRC analytics site per se shows what we have. Galaxy provides the analysis platform, and tools like HiFi and its DataMonkey incarnation are very good for performing surveillance, such as in the case of, for example, of the Hantavirus outbreak. Next slide, please. So since we don't have a lot of time today we're going to talk about results from the sort of end of the previous slide. I'm going to just highlight some of the analysis of evolutionary constraint that we did with DataMonkey. But I want to say that in our case, all three resources have MCP servers. So it's really no point in designing, kind of a pathways for visualizations or having actually services of any kind, because all of this can be just driven from AI agents. For Galaxy and for this entire analysis that I'm going to show, we are going to be releasing an AI agent next week. It's called Orbit, because we're sort of keeping it in this galactic environment. And because we have Galaxy MCP, we have BRC MCP, and we have DataMonkey MCP, this can all be done in a very transparent way, just from the prompt. So DataMonkey is evolving as well. There was the previous version, the server side, but we have the client side where for small data sets, everything is computed in your browser. Of course, there is a degree of to what extent you can do things in your browser. And then if you have large data sets, and it switches to a server-side execution. So let me close the door. Okay, next slide, please. So I already mentioned that we have MCP servers for everything, and you can check out one for DataMonkey. It's mcp.datamonkey.org. So it's basically imagine a situation where you have multiple alignments already, in which, so codon-aware multiple alignments for coding regions, you can just tell your agent, go look up DataMonkey MCP server and do these types of analysis that you would suggest. For example, you can completely replicate what we're going to be showing here if you have your own data. Next slide, please. So this is sort of a kind of the tools that DataMonkey provides for kind of for outbreak surveillance types of analysis. It includes recombination screening. It involves various tools for detecting selective pressure, both diversifying and for identifying selective constraints. So, and it sort of works at different scales. And in this slide in particular, so these 4 panels, they highlight the tools. So each of these things, like meme, for example, are the components of the DataMonkey suite that can be either accessed via the web or via MCP server, it doesn't matter. Next, please. So I don't know if I need to explain why evolutionary profiling matters, but of course, if we have, if we have enough outbreak surveillance data, we can say a lot of things, where things are going, which site may be important for downstream efforts to fight with these pathogens, and so on and so forth. So really, comparative genetics types of analysis is at the core of what we do with surveillance data. Next slide, please. You can actually tell a lot by looking at the DataMonkey logs in terms of who analyzes, where are the spikes of analysis. So for the Hantavirus, that's the timeline. And since I think, at least in the news cycle, Hantavirus is now eclipsed by Ebola, there is also a Ebola timeline. So what this shows, it shows spikes in the analysis on the DataMonkey platform. Next slide, please. So Hantavirus looks kind of a boring if you look at it from, I don't know, from 10,000 feet. If you look from 10,000 feet, there's not that much variation. And so in this case, we're looking at glycoprotein, which is in the M segment. It's a protein, I mean, previous speakers already described some of these things. It's a protein which is important for entry, and therefore it can be target for neutralizing antibodies. It also can reflect the host adaptations, and it's just generally good marker for outbreak tracking. So this tree, which you see here, it actually combines human and rodent-derived viruses. So you can see that, for example, the latest outbreak, so it's in the clade where you have rodent sequence as well. Next, please. So if you look at global selective pressures, this is very conserved. So you have GNDS on 0.02. So clearly, this is a very important piece of the virus. Let's move on to the next slide. But of course, things like GNDS, these are global measures that don't really tell you much, and you need to be able to look at the site-specific signatures. So there are these interesting sites, for example, which are indicative of host shift between human and rodent, for example. Next slide. You can also find sites which are potentially under diversifying selection, such as, again, slides that you see here. Next, please. You can also identify sites that co-evolve. And this is a very interesting analysis because in this context, non-synonymous sites are as important, synonymous sites are as important as non-synonymous sites. Because if you have covariation at synonymous sites that might indicative of some effects on RNA, for example. So really what these slides demonstrate, and the presentation, I sent this URL to everybody, so you can click on it and look in more detail. So these slides just highlight facets, different pieces of data monkey you can that you can use on that. Next slide, please. So in summary, this is the last slide of this presentation. So we can tell that this is it's very conserved. So again, as I mentioned, from bird's eye view, this looks boring, but we can actually identify sites which are unusually strongly conserved. And this is the least of these sites. This particular analysis was done with the components which listed at the bottom. And again, if you have sequences, you can use the components that I described for example, pulling glycoprotein coding regions, generating codon-aware alignments, feeding all of this to DataMonkey. So essentially the whole thing from sequencing reads to the inferences that I just showed. That's it.
I know that was a lot of different information from 3 very different resources, but if anyone has any questions, we're all happy to stay on for as long as you need. But thank you for joining us, and yeah.
And yes, it is all in this file, in this slide deck that I shared via the chat. So copy it now because the chat will disappear, of course.
I will put all the links from this presentation with the recording that will go up on our YouTube channel. So this is the link for the BRC Consortium YouTube channel that's in the chat now. And there's a playlist for Tools for Tomorrow that all the previous ones are recorded and uploaded there.
There appear to be questions in the chat. So what is the scope of tools on DataMonkey? Mainly phylogenetic or also tasks such as assembly. DataMonkey doesn't have assembly tools, but Galaxy does. So if you want to perform assembly, you can do that. What else here?
Thank you, everyone. You don't have to stay. So any other questions? Have a great day.